Good morning. It's a pleasure to be here to talk to you about this very exciting and intriguing aspect of modern biology. I'm grateful to Tom Check and HHMI for inviting me and my colleague Nadia Rosenthal to give these lectures. It's especially nice to be able to talk to high school students in particular because this area is just now beginning to blossom or burgeon. You'll see that there are very interesting problems, the answers to which we don't know. And I'm hoping that some of you will decide this is an area where you'd like to make a career and help us work on and solve these problems. Now, stem cells and cloning has been much in the news, of course, as you all know, and one of the goals of my talk is going to make sure that we understand the basic scientific facts. I'm going to begin, therefore, by talking about the basic biology of development, about how animals develop, how we come from a fertilized egg, because that sets the background for understanding the special properties of stem cells. We'll talk about stem cells, about how they're involved in regeneration, possibly involved in aging, and what is the role of cloning and cloning technology in all of these studies. Today then I'm going to begin in the first part with how we come from an egg. Some of this is intuitive, of course, that there's a lot of growth and development from a fertilized egg, but I'm going to review some of the basic biology of that, and then we'll move on to talk about stem cells. Now, there are two aspects to development. I'm going to use the example of human development, and this involves both growth and differentiation. A human egg is about the size of a period in a sentence, and obviously when it becomes a person like yourself, there's been a lot of growth involved. Now, that development then has these two aspects that we'll say something about. In the first case, we can just think a bit about the growth. The growth is very rapid during embryonic development, and then in the adult stage, it's rather slow. It's less growth and more just maintenance. A second aspect, of course, is that cells become different. As the embryo divides, there's a period when cells have to be set aside to make different parts of the body. Now, it wasn't always obvious that differentiation was a gradual process, and that's one of the points I'm going to make this morning. It was in fact thought for many years, for centuries, that development was just a consequence of growth, that there was no change in the cells. And I'm going to show you a funny picture, which is how people thought about development for many centuries. This is a picture of a little animal, a human in this case, called a homunculus, who was thought to be all folded up in the sperm head, as shown here, and that all that was involved in development was the growth of this. That is kind of like those little sponge toys you have as a child, where you get a little sponge and you put it in water and then bingo, something comes of it. That was what was thought as being the main mechanism of animal and plant development for centuries. As you well appreciate, there is this process of differentiation, cells becoming more special as development proceeds, and that's what we'll talk about now. Now, the end point of the differentiation is that there are many cell types in your body. The exact number isn't known, but there are hundreds of different kinds of cells in your body. In this slide here, we see an adult human in the middle, and around her is examples of a few kinds of cells. You can see a skin cell up at the top left. There's muscle, then blood cells, bone. I'll be talking quite a lot about the pancreas and a cell there called the beta cell. There's the liver, the intestine, and two examples of nerves. So how do these different cells arise? To introduce us to that, I'm going to show you a short video, and that video will show you that it's a gradual process and attend to a very important early stage when cells begin to become different. In this video, then, we'll look at the beginning of development, looking into the ovary at an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus, and then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days, about 5 days. At this stage, then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal, and the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the three beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes. As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns, and appendages start to bud out. You see the head forming and the eye. And then eventually we get a small embryo, and some months later, of course, this would be born as a young baby. Now, what you saw there is both the complicated movements and the growth of the embryo. And I want to focus in a bit on these early stages of the formation of the germ layers, as they're called, the first signs when cells are being told what they will become. As you saw in that picture, there were three germ layers. I'm not exactly sure how to explain or describe why they're called germ layers, but they're germ in the sense that these are like buds or something that will develop into something new as the animal proceeds. We can think of them as sort of the primary colors. They're not drawn here as primary colors, as red, blue, and yellow, but rather as blue, green, and yellow. And you can think of them as the subset, or the sort of a substratum which is used to make the animal. And one point I'd like to emphasize then is that the outer layer, the blue layer, the ectoderm, is going to give rise to the whole nervous system and the skin. The inner middle layer, called the mesoderm, will make the bone, blood, and muscle, like the heart muscle or the skeletal muscle, etc. And I'll spend a fair amount of time talking about the third layer, the endoderm, which makes the gut tube, running from the mouth to the anus. And what is formed from the gut tube are the lung, the liver, the pancreas, the stomach, the intestine, etc. And in the next movie, I'll show you, to emphasize this point, you'll see that at the end, we begin to focus in on the development of the pancreas as one example. So if I could have the next animation, please. So here we're going to go through quickly the early stages of development again. And then we're going to get to this stage when the germ layers are set aside. The three germ layers, the ectoderm, mesoderm, and endoderm. So here you'll see then that the inner cell mass gives rise to the entire adult animal, in this case a human. This same process would occur in a mouse, as we'll see a bit later. If we now look at the embryo after the three germ layers have formed, this video will highlight what comes from the blue germ layer, the ectoderm. You'll see that it makes the nervous system, including the brain, and the skin. The middle part, the mesoderm, green here, gives rise to the muscle, including the kidneys, the heart, and the endoderm gives rise to the whole gut tube. There you see the lung, the liver, the intestine. And to give one more detailed example of this, let's think about the development of the endoderm, and in this case, the formation of the pancreas. So there's the pancreatic bud, which comes out of the endodermal derivative. Now I'm going to talk to you a bit today about the pancreas as one example of how an organ and then the cell types within that organ get made. And in order to do that, I have to remind you a bit about what the pancreas does. I doubt any of you would say that the pancreas is one of your favorite organs or that you even think much about it. But I'll remind you that when you had breakfast this morning, you did that to gain nourishment, and you couldn't do that without the important role of the pancreas. So where is the pancreas, and what does it do? Let's have a look here at our little human model. And the pancreas is a banana-sized organ found right next to your stomach and your intestine. So I'm going to remove the stomach here from this little fellow. And you can see this white organ here, which is the pancreas. As I said, it's about the size of a banana. And in the next slide, I show a picture of what it looks like. That's here. So you can see it's nestled in next to the top part of the intestine. And the reason is that it serves its physiological function related to the digestion and the use of food. That purple line there is a duct into which are secreted all the enzymes that the pancreas makes that allow your intestine to digest the food that you've eaten. And we can see that in a bit more detail here as to what at the cellular level of this tissue or organ is involved in digestion. You can see here a cell called an exocrine cell, which is the cell that makes these enzymes. They're secreted then into that inner tube that flow into the intestine to digest food, as I've said. There's a blood vessel right next to the endocrine function of this organ, the endocrine component, which is called an islet, standing for island. And I want to look a little more detail at this pancreatic islet, because within it, it has just 4 kinds of cells. And an important one that I'm going to use as an example of cell differentiation is called the beta cell, shown in yellow at top. And the beta cell makes the hormone insulin, which many of you will have heard of. The other 3 cell types make different hormones, like glucagon and somatostatin. Now I want to show you a real-life picture of this, which is actually a stained picture of an islet, which is done with a microscope that measures the fluorescence of the stain, called a confocal microscope. And so while that cartoon there gives you an idea of what it looks like, in real life an islet looks like this. In this case, the colors are a little different. The insulin-producing cells are in blue. Now, why am I so obsessed with this pancreatic islet? Why have I chosen this as an example? I could have, of course, chosen any example, a cell in your eye, a cell in your heart, a muscle cell, or a blood cell as an example of differentiation. But one of the important reasons I've chosen this one is that the production of insulin is very important in order for your body to make use of the food you eat. And once the sugar enters your blood, once your food is digested into sugar and it's flowing through the bloodstream, the islet measures the amount of sugar in the blood and then secretes this hormone, insulin, in order for the rest of your cells to take up sugar and use it. Without insulin, one doesn't survive. And there are 2 diseases related to insulin and sugar utilization. They're both called diabetes, type 1 and type 2 diabetes, and I'll say a bit more about those today and tomorrow. But for now, I want to make the point that the pancreas is very important in terms of thinking about medical problems like diabetes, in addition to understanding normal development. So let's talk a bit about normal development here. That is, how does the body make a cell type like this? As I've already said, this doesn't just happen like magic, going from a completely unspecialized cell in order to a fully specialized pancreatic beta cell, but it's rather a stepwise process. And in this slide here, we can see some of the steps involved in that process. You'll remember in the early development, I showed a stage called the blastocyst, inside of which there was the blue cells of the inner cell mass. So one of those is depicted here up at the top, ICM. And that cell then makes decisions about becoming, or some of its daughter cells make decisions to first become endoderm, then a pancreatic bud cell, then an endocrine cell, which is the islet component, and then finally within the islet it's going to be a beta cell or some other type of cell as shown here. Now, in order to really understand this process, we need to know what's going on at each of these steps. And as you will appreciate, this involves turning genes on and off, which make your cells different. The reasons your lens cells of your eye, for example, aren't like a pancreatic beta cell is because the cells in the eye express lens crystalline, and a pancreatic beta cell expresses insulin. So they're expressing different genes. Now, as scientists, we can tell what genes are being turned on or off by using a technique that involves something commonly called a DNA chip. This is a small microscope slide onto which has been spotted all of the genes, a copy of all of the genes in the human genome, which is about 30,000 genes. And so I'll show you with an example using a nerve cell here how we would go about doing that. So if we wanted to know which genes were expressed in this nerve, so here we have the head of the nerve where the nucleus is found, and here's the axon, and then here's the business end that sends the signal. If we wanted to know what was going on in terms of which genes were on and off in this, we would extract the nucleic acid, which I'm going to do here with this turkey baster since we've just finished Thanksgiving. So you'd suck out the nucleic acid, extract it from the cell, and then using the part of the nucleic acid called RNA, which represents the genes which have been expressed or turned on, one can make a probe from that and label the probe with a fluorescent marker. That's then applied to a DNA chip, and I have my little DNA chip here, which some of you may remember from playing games as a child. We then take our labeled probe and put it into this chip like this, and this will, because there are spots representing every gene in the genome, tell us exactly which genes are on, shown here, or off in the other color. Now I've done this with a nerve, and it would be a different result, a different pattern if we used a different cell type. And I'll show you in the next slide an example of a chip, a cartoon of one, and what it would look like. So here in principle then are the 30,000 genes found on the chip, and some of them will be what are called cell type specific, like the nerve I just did, and others would be genes found in all cells, sometimes called housekeeping genes. Things like histones, the gene for the histone protein would be expressed because all cells need histones to wrap up their chromosomes, or there would be other proteins needed for this general cellular machinery. Here's then an example to make or emphasize this point that different cells express different sets of genes. And if I did my little trick right, you'll see that our connectoid there looks just like a nerve cell. Blood cells would have a different pattern, as would a pancreatic beta cell. So the point here then is that we can use the expression of genes to monitor what's happening during development as cells are making decisions about their fate. Now, how do we put this all together? Well, if we want to know what happens during the life of a cell, how do you get from an unspecialized, undifferentiated embryonic cell, like an inner cell mast cell, to a pancreatic beta cell? At each step, genes are turned on and off. And we can see that here in this slide. The inner cell mast cell then has the decision to make initially, should it be part of the ectoderm in blue, mesoderm in green, or endoderm in yellow. That sort of lineage tree might remind you of your own ancestry, trying to think of where things come from. And similarly, those kind of lineage studies can be done on a cell like this. The first decision it's make in this picture is to become an endoderm cell, turning on some genes. The next decision is within the endoderm, not to be lung, not to be liver, but instead to be pancreas. Within the pancreas, it's making a decision to become part of the endocrine component, the component that will make hormones. And finally, it makes a decision to become a pancreatic beta cell. So here, if you like, is the sort of molecular history of what a beta cell looks like. And I'd like you to think about an analogy that this relates to how your own life might unfold. What are the decisions involved in your life to tell you to become, say, a soccer player or a lawyer or a scientist? There are all kinds of influences that impinge on those decisions. And it's not a process where when you arrive from your mother's womb, you decided right then to be a scientist. This is a stepwise process that involves your education. You're affected by your neighbors in school, by your parents, and by others. And similarly, cells have to make these decisions by influences both internal and external. And so I'm going to now move to this point of how do cells know what genes to turn on and off? What are the signals that tell a cell what to do? And in general, there are two kinds of signals, one internal and the other external. The internal signals, which happen earliest in development, come from cytoplasmic factors. These are factors which are in the cytoplasm of the egg and begin by sending signals into the nucleus, usually by a transcription factor, to tell it which gene to turn on and off. And we have a little video, if I could have that next, to show you what that would look like. So here you see these factors. They're color-coded to correspond to the germ layers. And they're going in and out of the nucleus to tell that nucleus which genes it should turn on and off, for example, to initiate development. We'll now move to the other kind of signal. So as initially cells are being told which part of the germ layer, which germ layer they should pick. And then they're going to move to the issue of signals by adjacent cells. Here are adjacent cells that are involved in determining cell fates. In this case, I'm showing the example from the endoderm, where adjacent cells, either other endodermal cells or cells from the mesenchyme, which is a mesodermal derivative, send a signal. And as you see here, these signals go back and forth, telling the cell what it should do. And then once it makes a decision, it will send signals back to the signaling cell. That can then result in the end of the cell making the decision to become a pancreatic bud cell. Well, I've talked about these signals in a general sense. What are they in particular? They are almost always a kind of gene product called a growth factor. Now, that's a little bit confusing because I'm telling you that they're not involved in telling the cells to grow, but they're called growth factors because they were initially discovered by their ability to make cells grow in a tissue culture dish. There are families of growth factor molecules, and there are 100 or 200 important such signals known in development. What this looks like in this picture here is that a neighboring cell will synthesize a growth factor, here shown as a little circle or a polygon, and then it gets secreted by that cell to its neighboring cell. And when the neighboring cell receives it through a protein receptor, a transmembrane receptor on its surface, it changes the fate of the cell and tells it to become a pancreatic bud cell. I will show you in a few minutes examples of these signals telling stem cells what to do. But for now, I want to summarize at this point and say, what have we covered so far? We've covered the point that from a fertilized egg to a full adult differentiated animal involves a multi-step process, a gradual, stepwise signaling to cells to tell them what their fates should be.